Okay, welcome, my dear friends. CPGET 2023 free online coaching and also JL, DL, all competitive exams in plant sciences. Our eminent botanists, especially from government degree colleges, they are delivering lectures for the young aspirants, those who are going to attend all these competitive exams. Because of this is the competitive era, my dear friends. So in this regard, today's our eminent resource person, Dr.G.Bhageerath Garu, Assistant Professor, Department of Botany, Government Degree College, Rangasaipeta. Sir is going to explain about the genetics, especially one of the most important area for all competitive exams, including CSIR NET, AP and TS sets, all competitive exams like PG entrance and B.Ed. So this is our most important, even the JL, DL examination. So Sir has delivered his lecture for our young aspirants. It's valuable time for us. So better utilize this wonderful opportunity, my dear friends. So we would like to invite today's our eminent resource person, Dr. Bhageerath Garu. Please welcome and share your screen and start your session. I have given to you already. Welcome, sir.
Thank you, sir. Thank you very much. I'm very happy to associate in this CPGET coaching for the those who are interested to take admission within soon in the next academic year, that is PG in Botany. So I'm very much thankful to you, organizers, and as well as especially PADALA sir. Okay. I will share my screen. Is it visible, sir? Screen is visible to you?
No, sir. Please click on please double click on the PDF.
Once again, today's is our topic in this online CPGET coaching in botany subject, that is structural and numerical variations in chromosomes and their applications. So chromosomes are nothing but it is vehicles of heredity containing many number of genes. At the time of cell division, these chromosomes are undergo some changes. These changes resulting in phenotypic or some traits or characters are change in the next generations. So that's why what are the major changes generally found in the chromosome structure, whether it is a minute change sometimes or maybe very large change, we can also found in the chromosomes. So these chromosomal aberrations or chromosomal mutations are classified into two types. One is structural changes in chromosomes. Another aspect is numerical changes in the chromosomes. We can discuss today two aspects of the chromosomal mutations. One kind of mutations occurs in the chromosome, which are visible changes in structure of chromosomes, which involving the total number of genes, or sometimes the arrangement of the genes also change. So that means what happened before cell division, the chromosome containing this type of shape or structure. After completion of the division, in the next generation, the chromosome shape may be changed like this. So this chromosome size is large initially, but later on the chromosome size is decreased. How it is possible? So what are the changes takes place in this chromosome? sometimes this chromosome is as usual like previous model, but the number of genes which are present on the chromosome, the positions are changes. So the order of the genes are changed sometimes, or sometimes addition of the genes, sometimes deletion of the genes. So that means the structural changes in the chromosomes associate with whether total number of genes are changed, or sometimes the number of genes present on the chromosome is rearranged. So these two kinds of changes are seen in the chromosomes at the time of cell division. So the structural changes are divided into 4 types. The first one is deletion, second one is duplications, third one is translocations, and the fourth one is inversion. So these chromosomal aberrations are broadly classified into 4 types. The first two types are deletion and duplications, which involved in number of genes loci. So the number of genes are changed after completion of the division process. So number of genes change means in any chromosome, loss of genes or sometimes add of genes are takes place. Loss means కొన్ని జన్యువులను కోల్పోతాయి. Another chromosome receive more number of genes. So this kind of changes comes under deletion and as well as duplications. These two type of chromosomal aberrations are involves in number of genes loci. So remaining 2 kinds of aberrations, that is translocations and inversions. These 2 kinds of chromosomal changes involve in order of genes are changed. What is that? Order of genes is changed. So order of genes is changed in translocations and inversion. And in the first 2 cases, number of genes are changed in the gene loci. So broadly, you can remember this difference between deletions, duplication, one set, another set translocation, inversion. So these 4 kind of aberrations related to the structure of chromosome. In the cell biology, you already studied the chromosomes are mainly observed in different shapes: metacentric, submetacentric, telocentric, acrocentric. So the shape maybe sometimes changes due to the loss or addition of the genes, or exchange or replacement of the chromatid segments. So these are the 4 type of chromosomal aberrations we discussed first. Later, we go for numerical changes. Are you followed? Hello?
Yes, sir.
Okay. Next, we can go for next slide. So what are chromosomes? You know very well. Chromosomes are the structures that will hold the genes in the form of genetic material, that is DNA. And the most eukaryotic chromosomes, including packaging proteins called as histones, bind and condense the DNA molecule to maintain its integrity. So every chromosome have 2 segments called 2 arms. These 2 arms are connected with a small constricted area, a spherical part that is known as centromere. Either side of the centromere, we can see 2 arms, that is P arm and Q arm. Generally, it is called as like this. So we can go for first structural changes. So any change which alter the basic chromosome structure is known as structural change. So chromosomes are already we can explain, that is vehicles of heredity, heredity material or genes. Any alteration, any addition or any deletion of chromosomal part leads to alteration of number, position and sequence of the genes in the chromosome. So first we can go for deletion. What is that? Deletions. Deletion is nothing but Deletion is nothing but a small segment or a portion of chromatid loss from a chromosomal complement or a haploid set of chromosomes. A portion of chromosome is missing or deleted, the chromosome becomes shorter due to loss of one or more genes. So for example, this deletions also sometimes called as deficiency. What is that? Deficiency. So for example, a chromosome is there. The terminal part or apical part of the chromosome, the terminal or apical part of the chromosome loss, it resulting the chromosome become very short. So this is nothing but is called the terminal part of the chromatid removed from the chromosome by breaking, by one break. This is nothing but is called as deficiency. When we can call as deletion, sometimes a interstitial segment or intercalary segment. Intercalary segment means this part of the chromatid removed from the chromosome by how many breaks? It requires how many breaks? An intercalary segment of the chromosome detached from the complement. So it requires how many breaks? Two breaks. But in case of deficiency, one break is enough to remove the terminal part of the chromatid. So in this case, what happened? Just you can observe. So the chromosomal complement, haploid set is there, no? In one chromosome, a small portion is deleted. So the deleted part is a loop-like structure, in the homologous chromosome. For example, this part will be removed in one chromosome only. The another homologous chromosome is there. It shows that part is formed a bubble-like projection. A bubble-like projection is formed in another homologous chromosome due to missing the homologous part. దీనికి సంబంధించిన homologous part ఇక్కడ లేదు. ఏమైంది ఇక్కడ? ఇది delete అయిపోయింది. Delete అయిపోయింది కాబట్టి that part form a bubble-like portion that is known as loop. What is that? Loop. So deletions are two types. One is deficiency, terminal deficiency or terminal deletion. Second one is intercalary deletion. Intercalary deletion means, see here, this is the bubble-like portion formed in the homologous, because here CD is missing. The corresponding homologous part is missing. That's why in one chromosome, it shows a bubble-like projection is formed. It indicates that the homologous part is missing in another chromosome. So this is called as interstitial deletion or intercalary deletion. Are you followed? So this deletion may occur at any time of during the cell cycle. Maybe it occur in the germ cells or somatic cells. If due to the deletion, one chromosome lost genes. One chromosome lost some genes. Maybe it is a single gene, sometimes of a block of containing several genes also. కొన్నిసార్లు ఒకే జన్యువును కోల్పోవచ్చు, కొన్ని సందర్భాల్లో ఎక్కువ జన్యువులను కూడా కోల్పోవచ్చు. So the deletion is one of the structural changes in the chromosomes. So just you can observe two examples are there. One is Cry-Du-Chart syndrome. What is that? Cry-Du-Chart syndrome is a good example. Cry-Du-Chart syndrome is nothing but a small short arm missing in the 5th chromosome of human beings. Which chromosome? 5th chromosome of human beings. A small short arm is missing. So due to this part missing in the 5th chromosome, the person shows one genetical disorder that is called as Cry-Du-Chart syndrome. So they cry like cat-like syndrome. Then a cat cry syndrome. So the infants are crying like a cat. So this type of syndrome we can see due to the resulting of what? Deletion. What is that? Deletion. Another one is Philadelphia chromosome. The Philadelphia chromosome disorder observed in the 22nd chromosome. So in this chromosome, long arm is missing. So due to that, CML, chronic myeloid leukemia. Chronic myeloid leukemia cancer cells are formed due to the loss of the many genes in the 22nd chromosome. So this is about deletions. Are you followed? Are you understood about deletion or deficiency?
Yes, sir.
Yes. Next, we can go for duplications. Duplications means the presence of few genes occurs more than once in a haploid complement. The presence of few genes occurs more than once in a haploid complement is called as duplications. దీన్నే మనం తెలుగులో ఏమంటాం ద్విగుణీకరణాలు అంటాం. A portion of the chromosome is duplicated, resulting in extra genetic material. That means, for example, you can take one chromosome material. So this chromosome containing few genes. So for example, here AB is gene. You can see same type of genes again in the chromosome. Here also we can see AB. So a block of genes present more than once. A block of genes present more than once in a haploid complement. Haploid complement means two chromosomes are there. So in one of the chromosome, the genes which present more than once, that is called as duplications. What is that? Duplications are there. So duplications again divided into three types: tandem duplications, reverse tandem duplications, and displaced duplications. So in this case, duplications resulting what? One of the chromosome containing many genes, another chromosome loss. Another chromosome loss of genes takes place. So deletion and duplication both are involved in the total number of genes loci on the chromosome is changes. So first we can go for tandem duplications. What is that? Tandem duplication. Tandem duplication means you can refer any textbook in the in your whether it is Telugu Academy or another textbook. So for example, ABC is there and DEF is there. So the DEF genes repeated more than once. So DEF, the series of genes present in continuation with same order. So that is called as what is that? Duplication. Duplications means any chromosome containing a block of genes repeatedly repeats on the same chromatid. So that is nothing but is called tandem duplication. అంటే ఏబిసి ఉందనుకోండి, మళ్ళీ ఏబిసి జీన్స్ కనుక ప్రక్కన ఉన్నట్లయితే మనం దాన్ని tandem duplication అంటాం. ఏబిసి again ఏబిసి. So these ABC genes are carried by another chromosome, another homologous chromosomes. So that's why this chromosome containing many genes in another homologous chromosomes loss of some genes. So this is called tandem duplication. Reverse tandem means what? So if ABC genes are there, the same repeat, the additional block of genes are called as repeat. The additional block of genes are called as what? Repeat. So these repeated genes are arranged in reverse order. Arranged in reverse order means what? C, B, A. So this is called as reverse tandem. What is that? Reverse tandem duplication. Tandem duplication and reverse tandem duplication means the block of genes are similar order in the tandem duplication, but the repeated block of genes in reverse order. Here ABC is there, but reverse order is what? CBA. So this is called as what? Reverse tandem. You see these two examples. So here ABCD and here see ABC, DEF, GH. So ABC, DE and DE. So both the genes are there. Here CDE, CDE. So EDC, reverse order. So that's why it is reverse order. So if a block of genes received by a non-homologous chromosome, that is called as displaced duplication. Displaced means the repeated block of genes received by a non-homologous chromosome, LMN is there. So this is another chromosome. This is ABC is there. So it is received by a segment of the additional block of genes received by non-homologous chromosome, that is called as what is that? Displaced duplications. Displaced duplications. Duplications are three types. One is tandem and a reverse tandem and displaced. See here MNOPCD. So here this chromosome receive CDE genes. So this is called as displaced translocation. Sorry, duplications. Displaced duplications. So generally duplications takes place in many organisms more frequently, but they are less deleterious, less harmful, which helps in the formation of new gametes and as well as new species also. So this is about duplications. Are you followed? This is clear?
Yes, sir. Yes, sir.
Okay, next. So translocation. Translocation means a segment or a portion of chromosome transferred from one linkage group to another linkage group. A portion or a segment of chromatid or chromosome is transferred from one linkage group to another linkage group. One linkage group to another linkage group means what? In the linkage class, I explained that number of linkage groups in any organism that is equal to haploid set of number. Haploid set of number. So N, this is the value of haploid set chromosome number, is equal to linkage group. Linkage group. So just we see this is one homologous chromosome. This is another non-homologous chromosomes. So the corresponding homologous chromosome containing same genes, but here this is a different homologous chromosome. That's why it is a non-homologous chromosome. So what happened? These translocations and inversions containing same number of genes. There is no change after modification. Here 100 genes are there present in 2 chromosomes. Later also how many genes are there? 100 is there. But the position is different. The arrangement is different. The position is different and arrangement is different. In previous case, deficiency and deletion, duplications, what happened? One of the chromosome containing many genes, another chromosome lost some genes. Are you followed? Here the chromosome containing same number of genes. But what happened here? So the arrangement and a sequence of the genes is changed. So see here. So this is again divided into 3 types. Simple translocation, reciprocal translocation, and shifts also. So simple translocation means segment from one chromosome will break and attach it to the another chromosome. See here, exactly in between C and D, the chromosome breaks takes place and it attached. Where it is attached? At the terminal end part of the chromosome. So that's why after change, ABC is attached with LMNOPQRES. So the corresponding homologous chromosome gene number is less and it containing more number of. Altogether is gene number is same. Altogether you can say the gene number is same. So it requires how many breaks? One break to cut the segment from one homologous chromosome and it is attached to the exactly terminal part. So one break is enough for a simple translocation. In the reciprocal translocation, it requires 2 breaks in one chromosome one break in another non-homologous chromosomes. So that's why DE part will be enter into the another non-homologous chromosome. So it requires 2 breaks in one chromosome, 1 break in another non-homologous chromosome. The genes are altered or changed. This is a set of chromosomes. So in the translocations, a segment of the chromatic or chromosome is transferred from one linkage group to another linkage group. So reciprocal means both are exchanged similar fashion. So see here, ABC portion and replaced with LMN. So that's why we are getting heterozygotes are formed. Sometimes non-heterozygotes translocations also formed. So this is translocations. వీటిని మనం ఏమంటాము ఫస్ట్ వాటిని విలోమాలు అంట సారీ లోపాలు, సెకండ్ వన్ ఇస్ ద్విగుణీకరణాలు. సో వీటిని ట్రాన్స్లోకేషన్స్ వ్యతికరణాలు అంటారు తెలుగులో అయితే. సో ఓకే, దిస్ ఇస్ ద థర్డ్ టైప్ ఆఫ్ క్రోమోజోమల్ అబరేషన్స్. ఇస్ ఇట్ క్లియర్?
Yes, sir.
Okay. So another one is a Robertsonian translocation. In this case, this is the normal chromosome. This is the non-homologous acrocentric chromosome. What is this? Acrocentric chromosome. So in this place, it will be cut and this part will be reunion to this non-homologous chromosome. This type of a special rare translocation seen in some animals and plants. So in a Robertsonian translocation, an entire chromosome arm has attached to another at the centromere. Exactly centromere they're going to attach. Loss of chromosomal segment, a few usually lost. It is a special case, but some of the textbooks it's not mentioned. So this is translocations. Inversions. The fourth type of chromosomal aberrations are called as inversions. Inversion means a portion of chromosome re-altered. A portion of chromosome or sequence of genes in a chromosome re-altered by rotating 180 degrees. By rotating 180 degrees. How? Just see here. So this is the chromosome. It's centromere. This chromosome rotate 180 degrees like this. 180 degrees. So on either sides, it will be broken and reattached. So this type of chromosomal aberration is called as inversion. A portion of the chromosome has broken off, turned upside down and reattached. As a result, the genetic material is inverted. So an inversion is produced when there are 2 breaks in a chromosome and the intercalary segments reunites in reverse order due to the segment rotate by 180 degrees Celsius. So see here, the inverted segment sometimes containing centromere, sometimes not. So the inverted segment containing centromere, this type of inversion is called as pericentric. What is that? Pericentric. Pericentric. So for example here, A, B, C, D, E, F, G, H, I. This is the sequence before inversion. So what happened when inverted along with centromere by rotation of 180 degrees, the gene order is like this. A, B, it will be attached here. So E, G, D, C, H, I. So this is the after inversion. You can see the gene order is completely changed due to the break in 2 points and reunited at that point. So inversion of 2 types, pericentric inversion, paracentric inversion. Pericentric means if the inverted segment includes the centromere. Here centromere is present. This is called as pericentric inversion. If the centromere is absent in the inverted segment, that is called as paracentric. So these inversions and translocations are generally suppress the crossing over. So once you can observe what is the difference between the translocation and crossing over. So translocation we already discussed. Generally, the segments are exchange takes place between the homologous chromosomes. That is the crossing over. This is the two chromatids are there. Another homologous chromosome. So see here. So this is exchange of the two non-sister chromatids within the homologous chromosome. But translocation not like that. So this is the one chromosome. The another chromosome is not homologous chromosome. It is non-homologous chromosomes. That's why the translocation, the shifting of genes or rearrangement or exchange takes place between non-homologous chromosomes. So it is translocation. This is called crossing over. So just you have an idea what is the difference, how crossing over is different from translocations. So by here a pair of chromosomes which belongs to two different, and a pair of chromosomes which are belongs to same. So that is the difference you have to understood and remember for everything. So next, this inversions changes chromosome shapes also. How? For example, a V-shaped chromosome is there. It involves in the pericentric chromosome. It involves in the pericentric chromosome. It may be converted into hook shape. What is that? Hook shape model. So this is the shape of the chromosome after pericentric inversion. So that is the thing we can observe. Sometimes this vice versa also. So maybe a hook shaped chromosome is converted into V-shape. V-shape chromosome is converted into hook shape model. So these are the visible changes in the structure of chromosomes at the time of cell division. So inversion and translocations are both very important, which play an important role in the origin of new species. Due to the order of the genes are changed, it form different type of gametes at the time of meiosis, and which play a very important role. In these two chromosomal aberrations, we can see position effect. What is meant by position effect? So for example, before chromosomal aberrations, the position of the A gene is here. After chromosomal aberration, the gene position is changed into another locus. If it changes into the another locus, the action of the gene also influenced. Action of the gene is also influenced. At this place, this gene action is different after it separate and reunion with other genes. So the action of the genes is scrupulously changed when its position is modified due to the chromosomal aberrations like inversion and as well as translocations. So this is called as what? Position effect. What is that? It is called as position effect. Position effect ante chromosome lo జన్యు యొక్క స్థానం అనేది మొదటిగా ఉన్నట్లుగా కాకుండా మారినట్లయితే, దాని స్థానం మార్చినట్లయితే దాని యొక్క విధి, దాని యొక్క క్రియ అనేది కూడా ప్రీవియస్ మోడల్ గా ఉండకపోవచ్చు. ఇట్ షోస్ ఏ డిఫరెంట్ యాక్షన్. సో దట్స్ వై ఇట్ ఈస్ కాల్డ్ యాజ్ పొజిషన్ ఎఫెక్ట్. Position effect generally seen in translocation and as well as inversion. Are you followed?
Yes, sir.
Okay. So this is about structural changes in the chromosomes. Next, we can go for numerical changes in the chromosomes. So numerical changes means it related to the chromosome number only. In the earlier case, we discussed in the structure, but now we can discuss with number. Normally, every organism is diploid. They possess two sets of chromosomes. Any deviation, any change from the diploid state represents a numerical chromosomal aberration which often referred as heteroploidy. So, or we can change it is called a numerical changes in chromosome number or numerical variations. So, heteroploidy is mainly divided into two types, aneuploidy and euploidy. Aneuploidy and euploidy. So, just we can go for aneuploidy. Aneuploidy means any addition or deletion, one or few chromosomes from the diploid set of chromosomes. From the diploid set of chromosomes. So, for example, a diploid chromosome set containing 2x is equal to 10 at the time of meiosis. It produces two gametes, x is equal to 5. Another gamete is x is equal to 5. But due to some mistakes in the cell division, due to the effect of some drugs or chemicals or radiations, it involves in changes in the chromosome by abnormal behavior of the chromosomes at the time of cell division process. So, due to the chromosomal non-disjunction, chromosomal non-disjunction means at the time of meiosis, the two daughter cells received equal number of chromosomes. But sometimes what happened, the chromosomal fibers may not effective results. It receives an extra chromosome. It becomes as x is equal to 6. It lost one chromosome in one gamete. It becomes x is equal to 4. So what type of gametes are formed due to the non-disjunction of the chromosome? N plus 1 gamete. And what is that? N minus 1 gamete. Ante one chromosome is lost here. Here one chromosome is extra in the gamete. So this N plus 1 gamete at the time of fertilization, it fertilized with normal gamete. What you are expecting in the next generation? The cells containing 2n plus 1. This n minus 1 gamete fertilized with normal gamete. What you are expecting in the next progeny? The cells containing 2n minus 1. Are you getting my point? So due to the non-disjunction of the chromosome, what type of aberration, what type of status we can see? Aneuploidy. Aneuploidy means vishamasthitikalu antaru. Aneuploidy changes in chromosome number do not involve the whole genome. Either may be addition or deletion of one chromosome or two chromosomes or sometimes 3 chromosomes also diploid set. So these aneuploidy divided into two types, hypoploidy, hyperploidy. Hypoploidy means deletion. Hyperploidy means addition. Hypoploidy status arise due to this type of situation, due to this type of combination. When a gamete, n-1 gamete combined with normal gamete, we are getting hypoploidy condition. So if one chromosome is lost, that is nothing but monosomic. What is that? Monosomic. 2n-1 is called as monosomic condition. Two chromosomes are lost, 2n-2. That is called a complete set chromosome is absent in the organism. That is nullisomic. What is that? Nullism. It is very important. These 4 terms generally asked in PG entrance examination. Most any university which concentrate on this aneuploidy condition. So monosomy means from the diploid set of chromosomes, one of the chromosome occurs only once. ఒకటే ఉంటుందన్నమాట. దానికి సంబంధించిన corresponding homologous chromosome absent. So if total a pair of chromosome is absent among the diploid set, that is nothing but is called 2n-2, nullisomic. Are you followed?
Yes, sir.
Yes, sir. Yes, sir.
Okay. This is next one is hyperploidy. Hyperploidy means any organism, the cells showing a abnormal chromosome number to the normal ones. So a n+1 gamete fertilized with n gamete, we are getting this type of condition. That is trisomy, 2n+1, tetrasomic, 2n+2. And an addition of 2 chromosomes are there. An addition of 2 chromosomes along with diploid set. So addition of one chromosome along with the diploid set. So this is trisomic condition, tetrasomic condition, monosomic condition seen in different cases. You are studied in the genetical disorders in zoology subject. Down's syndrome, what is the example for which type of hyperploidy? Down's syndrome. 2n plus 1. 2n plus 1 example is Down syndrome. Down syndrome ఏమంటారంటే 21st trisomy అంటారు. ఏమంటారండి? What is that? 21st trisomic. 21st trisomic means a person who suffer with Down syndrome. How many 21 chromosomes are there in the cell? A1, B2, C3, D none. A person who suffering from Down syndrome, you can examine the cell containing 21st chromosomes. 1, 2, 3, none. నార్మల్ గా 21st క్రోమోజోమ్స్ ఎన్ని ఉంటాయి? అతనిలో నార్మల్ వ్యక్తిలో అయితే 2 ఉంటాయి. దట్ ఈస్ సెట్. అంతేనా లేదా? అనదర్ ఎక్స్ట్రా క్రోమోజోమ్ ఇస్ దేర్ 21. సో దిట్ ఈస్ ట్రైజోమిక్. వాట్ ఈస్ దట్? ట్రైజోమిక్. 21 chromosome, again 21 chromosome, another 21 chromosome. So that is 21st trisomy is nothing but example is 2n plus 1. That is trisomic. Are you followed?
Yes, sir.
Yes, like that. Klinefelter syndrome. Klinefelter syndrome, what you are expecting? How many chromosomes are there? As a person who suffering from Klinefelter syndrome, 47 chromosomes. But human beings normally containing how many chromosomes? 46. So kabatti just you can write Klinefelter syndrome is equal to 44 plus XXY. What is the additional chromosome is here? Which chromosome is extra here? X chromosome. So this is another example for this extra additional chromosome present. That is X chromosome is there. So that is Klinefelter syndrome. Turner syndrome is there. Turner syndrome is example for 44 plus X0. 44 plus X0. So X chromosome is missing. X chromosome missing in the females. That genetical disorder is called as what? Turner's syndrome. Turner's syndrome. In this way, many plants in Datura, in Inothera, plants are also seen. For example, double trisomic also is there. So double trisomic means 2n plus 1 and 2n plus 1. So this is 2 chromosomes extra additionally add to the normal diploid set. So this is called double trisomic. Double trisomic. It is actually tetrasomic, but 18th chromosome occurs 3 times and 12th chromosome is also occurs 3 times. And 2 different chromosomes occurs 3 times. That is nothing but 2n plus 1 plus 1. That is called double trisomic. What is that? Double trisomic. So in this way, you can list out some examples for each monosomic, nullisomic, trisomic, and tetrasomic. So it is an addition or deletion of one or few chromosomes from the diploid set of chromosomes confined as aneuploidy. Are followed? Is it clear?
Yes.
Okay, sir.
Okay. Next. So this is monosomy. అంటే ఇక్కడ ఏం మిస్ అయింది? Just see this karyotype. 7th chromosome is miss. 7 chromosome is missing. So that's why it comes under monosomic. Monosomic is formula 2n minus 1. Monosomic formula is 2n minus 1. అంటే ఒక క్రోమోజోమ్ ఒకటే ఉంటుంది. దానికి సంబంధించిన రెండవ జత ఉండదు. So that's why monosomic is nothing but 2n minus 1. You can confine like this. So next one is nullisomic. So here which pair of chromosome is missing? Which pair is missing? This karyotype containing 8 chromosomes. 2n is equal to 4. But nullisomic, how many pairs are there? 6. Which pair is missing? 4th pair is missing. So that's why it is the represented as
2n minus 2.
Yes, exactly correct. 2n minus 2. So in this way, you and based on the diagram also you can explain the answer. Based on the diagram or karyotypic picturization, we can explain whether it is what type of aneuploidy, nullisomic or monosomic or trisomic. The numerical changes in the chromosome number, we can go for aneuploidy. Aneuploidy means a loss of or addition of chromosome from a diploid set of chromosomes. So we already discussed tetrasomic, monosomic. Tetrasomic means this figure, karyotypic figure, says that the 8th chromosome is in 4 times. 4 times means this addition, these arrow, down arrow chromosomes are additionally added to the organism by at the time of fertilization. By this, a particular chromosome is represented in 4, does instead of normal 2. So this is the tetrasomic condition we can see. So another one is euploidy. Euploidy means, you know very well, the change in the number of sets of chromosomes in an organism. Normally, every organism possesses only 2 sets of chromosomes. That is nothing but diploid number is there, 2n. But at times, there is an addition or loss of complete 1 set, more than 1 set of chromosomes is observed. It is called as euploidy. Euploidy means if in the diploid set, 1 set is completely removed, definitely it containing only 1 set, that is monoploid. Mostly gametes are receiving only 1 set of chromosomes. So every gamete having only 1 set of chromosomes, that shows monoploidy. So generally monoploids are not fertile. It is sterile plants because they are unable to undergo meiosis process or meiosis division. It unable to produce the pollen grains or ovules. That's why the monoploidy plants are treated with colchicine. The chromosome doubling takes place. It becomes as diploids. In nature, the polyploids are very less when compared to the most of the plants. A organism or a plant having more than 2 normal sets of chromosomes, they are called as polyploids. So polyploids mean an additional set is added to the diploid. So one additional set of chromosome is added to the diploid, it becomes triploid, 3n. So 2n to 2n add 4n, that is tetraploid. So pentaploid, hexaploid, like this. So in addition, the changes in the sets of chromosomes, changes in the sets of chromosomes of an organism, that is become as polyploidy. So the polyploidy nature is very less in the nature. So just you can observe. So normal chromosome complements like this. Every chromosome having a set, 1 chromosome occurs 2 times, 2nd chromosome also occurs 2 times, like this. If monoploidy means it receives only 1 chromosome from each pair, but polyploidy is more than normal number of sets of chromosomes. Normally diploid containing 2 chromosomes of every one. So additional 1 chromosome is added to every set. So that's why 2n plus n, it becomes as triploid. 2n plus 2n, it becomes as tetraploid, like this. So these polyploids are very, very important in plant breeding or in the crop improvement purpose. So these polyploids again divided into 2 types, autopolyploids and allopolyploids. Autopolyploids means the set number of chromosomes belongs to same species. For example, a diploid chromosome having genome AA, it fertilized with a normal gamete, which is also containing a genome. So by the fertilization between 2n and a normal gamete. We are getting 3N. So this 3N organism containing similar type of genome, similar type of genes. So the triple A, again this treated with colchicine, it becomes as 6X, hexaploid. So but hexaploid, all sets of chromosomes containing similar type of genes, similar type of genome. So that's why they are called as autopolyploids. What is that? Autopolyploid. If this diploid organism, the diploid gamete, combines with a normal gamete which shown another genome. For example, instead of A, you can write C. Then we are getting triploid, but the genomes are different. AAC. So AAC is a what type of polyploid it is? Allopolyploid. So a polyploid containing a number of sets of chromosomes, but all are similar genome. So that is the autopolyploid. In the nature, autopolyploids are very rare, and these polyploids are useful to increase the production of, for example, tea. The tea powder we are extracted from leaves only. So if any coffee plant or tea plant, it become polyploid, we can made as a polyploid. The polyploid shows 3 centimeter square area of leaf produces. A normal diploid tea plant produces 1 centimeter square leaf. So which one is better? Which one is beneficial to us? 3 centimeter square leaf is beneficial because we are extract the tea powder from the leaf only. If leaf size increases, automatically yield will increase. So if the for example, sweet potato type tubers are there, the tubers containing number of sugars, the amount of sugar increases in the polyploids. If the tubers, for example, Pusa giant Napier grass is there. Pusa giant Napier grass is one of the polyploid. What is that? It is a feeder plant. Pashugrasanik sambanchina mokka. Pusa giant Napier grass is developed from the 2 different grasses by what is the Indian Council of Agriculture Research. So allopolyploids are those polyploids which contain 2 or more different genomes. So I am saying AAC, it is a different genomes are there. This become as a allopolyploid. Triple A is there. This is autopolyploid. So in this way, you can easily identify whether it is autopolyploid or allopolyploid. So common wheat, we are using in a nowadays wheat flour. It is a hexaploid. What is that? Hexaploid. This hexaploid containing 3 different genomes. Triticum, sickle cereal, rye and wheat plants are produced Triticum dicoccum. So this species is usually used in the our market for the wheat flour production. So గోధుమ పిండి ఎక్కువ తయారు కావడం కోసం ఒక hexaploid ని తయారు చేయడం జరిగింది అన్నమాట. So Raphano brassica, what is that? Raphano brassica is there. So it is also one of the allopolyploid. Raphano is Raphanus sativus, one plant, and it is crossed with Brassica oleracea racum capitita. So variety capitita. So cabbage and Raphanus rendu kaliste vachindi Raphano brassica. Triticale is also, it is a allopolyploid. So in the hybridization process, we can polyploids are made by a diploid gamete fertilized with normal gamete, or due to the failure of meiosis, 2 gametes are produces tetraploids, or sometimes any mutagens are there. These mutagens influence the arising of polyploid plants. So mostly polyploid plants are produce vegetative modified plants, which helps in the production of sugar in the tubers and increasing the leaf area. And whatever that vegetative propagated plants are generally produced by this polyploid. So this is things you can observe in your examples also quoted in your textbooks. So see this one, these are the two major kinds of polyploids in the chromosomal number based on that. So implications of chromosomal aberrations in plant breeding and crop improvement. Aneuploids have been used to determine the phenotypic effects, whether loss or gain of the different chromosomes. If one chromosome is lost, what type of disorders we have seen in different genetical syndromes. Aneuploids are also used to produce align addition or align substitution lines, which are useful in genetic transfer from one species to another. So aneuploids are useful to identification of chromosomes involved in translocation. So for example, in the chromosomal aberrations, for example, you can see if one gene is missing in one chromosome due to the deletion, the recessive gene expressed that character. For example, A gene is deleted from the chromosome, but here in another homologous chromosome containing small a gene. By missing this gene, it expresses its character easily. So this is called as pseudo-dominance. What is that? Pseudo-dominance. So this type of gene actions also we can know by this chromosomal aberrations. What is the function of the gene when absence of dominant gene? We can easily trace out by these chromosomal aberrations. So this is the important topic in the entrance point of view. So chromosomal aberrations with respect to structural changes as well as numerical changes. So mostly numerical changes nunchi questions are frequently asked. It is very easy to remember that what is meant by aneuploids and polyploids. So just you can go through the chromosome numbers very easily. 2n plus 1, 2n plus 2, 2n minus 1, 2n minus 2, nullisomic, monosomic, disomic, tetrasomic, like this. So this is the, today we discuss about this various types of chromosomal aberrations. So are you understood? I think.
Thank you, sir. Interesting.
Thank you.
Hello. Good evening. Good evening. How are you?
Okay, any doubts regarding of today's class, my dear friends? Dr. Bhageerath garu has clearly explained about the chromosomal
Yes, sir.
aberrations, and at the same time, autopolyploid, polyploids, autopolyploids and allopolyploids, okay, aneuploids. So these are all most important area, my dear friends. Okay, so really, thank you very much, sir. You have clearly explained about the ploidy and the chromosome structure and chromosomal aberrations. Thank you, sir. Any doubts?
No, sir. Thank you.
No. Right, sir. Thank you. Tomorrow we will meet.
Okay.
Right.
Thank you. Thank you.
Right, sir. Thank you.